The insects are unknown in their original or primitive manifestations, and consequently their ancestry is wrapped in obscurity. The subject can only be approached by the coordination of whatever evidence may be gleaned from three principal courts of appeal: comparative morphology, embryology, and paleontology. Of these, paleontology has, so far, remained almost like a closed book. No annectant forms bearing directly upon the problem have been disclosed in the strata of any geological period. Without this necessary record of the past, the links in the evolutionary chain are deductions based mainly upon morphology and development. For a considerable time past, the two most plausible theories of insect descent have been those which involved either the Crustacea or Symphyla as ancestors. The crustacean theory, which is based no external structure, is losing ground, while that involving the Symphyla has remained in much the same status that it acquired in the days of Packard. The most important evidence bearing upon insect ancestry that has come to hand during the last 30 years or more was Silvestri's one discovery, in 1903, of the remarkable Thassaneran Anogipix. Since that date, no revelation of comparable significance has been made. While this discovery added weight to the Symphylan theory, Packard's conclusion of 1898 too still holds good today. This writer stated that Scolopendrella is the only extant arthropod, which, with the sole exception of being progoniate, fulfills the conditions required an ancestor of the Thysanura and, through them, of the winged insects. In 1930, Tilliard III brought forward a theory which claimed that the insecta were derived from remote hypothetical nauplioid ancestors Protaptera. They arose separately from the Symphyla and other Myriapoda which, he maintained, had a similar origin. The Symphylan theory was rejected by this author owing to the old crux of the segmental location of the gonopore. The contention of the present communication is that the Symphylan theory is substantiated by a larger body of evidence than any other explanation of insect descent. In considering this theory, it needs to be stressed that it is generally accepted. The present article is an abbreviation of one bearing the same title: Trans. Society. Brit. Entom. 1, 1936, the main features of which formed the subject of a presidential address given to the Society for British Entomology, at a congress held in Cambridge, OU, June 26–29, 1936. Today that among the Thysanura are found the most generalized of all insects. Lepisma and its allies are the forerunners of the Pterygoda, while Anogipix and Campodia are more nearly related to the ancestral insects. The old phylogenetic importance of Campodia has now been largely eclipsed by Anogipix with its greater display of Symphylan characters. Since Anogipix provides an essential link in the present theory, these characters need to be recounted. They include simple multi-articulate antennae, styli and eversible sacs on most of the abdominal segments, cerci bearing the orifices of large posterior glands, mouth parts built on an essentially similar plan, an identical leg segmentation with single-jointed tarsi, a simple digestive canal with homologous salivary glands and the same fundamental type of excretory organs. A second link in the chain is provided by embryology. The lower insects pass through a developmental phase revealing 14 postcephalic segments, each bearing limb rudiments. This phase can only be interpreted as a relic of a former polypodous condition. On turning to the Symphyla, it will be noted that they reveal, in their adult structure, close resemblance in metamerism as evidenced by the appendages, namely, 12 pairs of legs, the 13th pair transformed into sensory processes and the 14th pair, as in insects, represented by cerci. Thus it seems very possible that the insecta recapitulate what may be regarded as a Symphylan stage in their development. In the derivation of the insecta from Symphylan ancestors, it would appear that three main evolutionary changes were involved. L. The development of a segmental tracheal system. 2. Concentration of the locomotory function in the first three postcephalic segments, with the consequent reduction of the appendages of the remaining segments other than cerci to a vestigial condition. 3. The development of posterior gonoducts opening between the 10th and 11th postcephalic segments, together with the atrophy of the original ducts. L and 2 require little comment since they are evidenced in the development of the lower insects and in the adult structure of the Thysanura. In connection with 3, it is noteworthy that a shifting backwards of the genital apertures during development is a common feature among many orders of insects. Furthermore, new or secondary genital passages N. Copyright 1937 Nature Publishing Group. 400 Nature March 6, 1937. Orifices have been acquired among Lepidoptera, Strepsiptera and in the Hemipterus family Simicidre. The postulation of a change from a progoniate to an opistogoniate condition refers, therefore, to the evolution of a class wherein diverse modifications of the genital conduits are a prominent feature. The insecta are peculiar in that the egg follicles are disposed in a linear series forming ovarioles, while at the apex of each ovariole is an end chamber or germarium containing the undifferentiated germ cells. In other arthropods no such arrangement into ovarioles prevails and the germarium is located parietally. It will be evident that structural modifications of the gonads must have been involved during the evolution of the insecta, and, it is claimed, that these changes occurred in association with the development of posterior gonoducts. So far, the Protura and Colembula have been omitted from discussion. They are side developments off the main line of insect descent. The Protura show a predominance of hexapod features, suggesting that the early evolution of this group was with the rest of the insecta. Afterwards they diverged as a specialized side branch but retained the primitive trait of anamorphosis. The Colembula display both myriapodin and insectin characters. Owing, possibly, to the paucity of yolk, which induced holoblastic segmentation, development became arrested and only 9 trunk segments formed. In the absence of ovarioles and retention of the parietal germarium, together with the general simplicity of the gonads, the Colembula betray close resemblances to the Symphyla. The possibility that traces of a former progoniate ancestry are revealed in Colembula, as mentioned by Claypole, needs further investigation. Evidence of such ancestry also requires exploration among other Apterygoda. Re-examination of the Symphylan theory seems, therefore, to establish it as the only adequate explanation of insect descent. In view of the many fundamental similarities between the Symphyla and Tbysanura, it is claimed that these two groups arose from a common stock, not very different from living forms of Symphyla. Theories involving separate origins for the two groups in question, on the grounds of disparity in the segmental locations of the gonopores, have to fall back upon the improbable hypothesis of convergence to account for their community of structure. If the propositions now advanced serve to focus attention upon aspects of the problem hitherto neglected, they will have fulfilled their purpose. 1 Ann. Della Scuola Sup. D. Oric. In Portici, Lit 1903, Ibid. 6 1905. Packard, Textbook of Entomology, 22 8. Roy. Sus. Tassum. 9. J. Morph. The Recent Floods 1 and the United States. By Dr. C.E.P. Brooks. Teehee, Mississippi and its tributaries together drain about half the area of the United States. The Mississippi itself runs through the center of the country, the Missouri rises in the Rocky Mountains and the Ohio in the Allegheny Mountains. The upper courses of the Mississippi and Missouri lie in relatively dry regions, where moreover the winter precipitation is mainly in the form of snow. The Ohio, on the other hand, runs through a region of comparatively heavy rainfall, and is much more liable to flooding. During the last few months of 1936, the rainfall in the valley of the Ohio and the Mississippi above Memphis had been abnormally heavy, the total for the months September-December being nearly 1.5 times the normal. Thus at the beginning of January, the ground was waterlogged and there was already minor flooding in some of the tributaries of the Ohio. In January. Paper read before the Royal Meteorological Society on February 17. A type of pressure distribution developed which was very favorable to prolonged heavy rain, figure I. Pressure was high off the east coast of the United States, and decreased steadily westwards. This was associated with a current of warm moist air blowing from the Caribbean Sea and the Gulf of Mexico across the eastern half of the United States. Above this warm and very moist southeasterly wind was an upper current from the west, which was relatively cool. The air was therefore highly unstable. Farther north, over the Great Lakes and southern Canada, pressure decreased northward, and the westerly current descended to ground level. The encounter of the warm moist southeast wind with this great barrier of colder air from the west gave all the conditions necessary for heavy and prolonged rain, which was localized by a series of depressions traveling from southwest to northeast along or near the line of the Ohio Valley. Earwigs insecta, Dermaptera, are characterized by uniquely elaborated cerci, commonly called forceps, the function of which remains unclear. The intrasexual and intersexual interactions in the laboratory to examine the context and pattern of forceps use in the toothed earwig. Bostock's Apictenlatus Codell. Interactions between pairs of earwigs were recorded in 4 social situations: 1 2 males, 2 2 males plus a virgin female, 3 2 females, and 4 1 male and 1 female. Forceps were used as both weapons and display structures by males and females in all of these social contexts. During pairwise male-male interactions, one male clearly dominated the other male. Dominant males were more active and more likely to use their forceps in intrasexual interactions than were subordinate males. In interactions where there were 2 males and 1 female present, the male that dominated male-male interactions was able to maintain exclusive access to the female. There was no indication of active female choice during or after courtship. During intersexual interactions, only males used their forceps during courtship. The behavioral repertoire involving forceps was greater for males than for females, especially in intrasexual contests. There was no clear outcome of intrasexual interactions among females. These results suggest that forceps function mainly as weapons in male-male interactions and may have evolved, at least in part, as a result of sexual selection. Further research is required to test for female mate choice and to separate the various mechanisms of sexual selection if mate choice exists. Comparative studies are needed to determine if sexual selection was the original evolutionary mechanism leading to the development of these unusual structures or if sexual selection is relegated to a secondary effect, leading to the elaboration and sexual dimorphism of these structures in selected groups of earwigs. Termite nest termitarium. The queen, the brood and most of the colony's individuals live in a so-called termitarium plural, termitaria. It is composed of mud that is sometimes as hard as concrete and a paper-like substance made from chewed wood. The conditions inside the nest are dark, moist and cool and suit the requirements of the mostly blind unpigmented termites with their soft cuticles. Runways or galleries are built by the workers radially from the nest in all directions and connect the termitarium with sites where the colony gathers food. Mound nest. These galleries are either subterranean about 20 to 50 centimeters deep or are built from mud packs or mud galleries attached to a stem or other substrates. The termites' road system can be enormous and reach a radius of 50 to 100 meters around the nest. Each termite species builds its particular type of nest, either a mound nest shown above made above the ground, or a subterranean nest in a buried log, a decaying stump or the crown root of a tree, or an arboreal nest attached to a branch or to the stem of a tree. Mound nests can reach a height of up to 6 meters. The mound nest of the Australian species Amyderms meridionalis is flattened and the tapered sides always orientate towards the north and south. Constructing the nest this way ensures that the inside of the nest is not heated up by the sun since only the narrow sides are exposed to direct sunlight. Subterranean nests buried at a depth of 50 centimeters or even reaching depths of several meters are invisible and therefore difficult to discover. Termite life cycle and caste system. The simplified model of a termite life cycle indicates the 3 castes, the reproductives, the soldiers and the workers. Due to the fact that termites are hemimetabolous insects, even the nymphs take part in the social life and have their specific tasks to fulfill. The so far poorly understood concept of caste determination does not seem to be definitive or too rigid. Once the caste of an individual is determined, development into other castes is still possible. Soldiers, also referred to as intercastes, might turn into workers or even into reproductives, if there is a shortage of individuals of other castes. This process is controlled by pheromones. In the case of the queen, there is a specific, queen, pheromone, preventing other individuals from turning into queens. Only if the queen is removed or dies, does the lack of the specific pheromone promote the development of a new queen. Reproductives possess compound eyes and are more or less brown due to their sclerotized cuticle. Developing reproductives have wing buds, wings or wing stumps. Reproductives can be further divided into Elates, the young winged reproductives of both sexes. From time to time about 100 to 1,000 elates leave the colony for a mating and colonizing flight. After mating, a pair settles down at a suitable site like a rotting scar on a tree in order to establish a new colony. Elates that cast their wings after the colonizing flight and successively turn into queens and kings. Initially only a few eggs are laid and brought up by a female alate. As the number of individuals in the colony grows, the more workers are available to help the young queen to care for the brood. After 3 to 5 years, the number of individuals is already so large that the colony of a pest species can turn into the damaging stage. Queen and king, which are the main reproductive individuals in a colony. Once there are many workers to help the queen, her only job is to produce a tremendous number of offspring. A large queen may lay more than 1,000 eggs per day. The lifespan of a queen can be as much as 50 years. Neotenics assist the queen in laying eggs once her productivity decreases. When the queen has died or deteriorated, one of the neotenics takes her place. That is the reason why the removal of a queen from her colony does not necessarily mean the end of the colony. Workers are sterile, wingless and blind males and females. Their cuticle is unpigmented and not hardened, therefore the animals are confined to a dark and moist environment. Workers build the nest in galleries, they fetch food, care for the brood and feed reproductives and soldiers. The worker's lifespan is 1 to 2 years. Soldiers are, like workers, sterile, wingless and blind males and females with an unpigmented, unsclerotized cuticle. Soldiers defend their colony from intruders by the use of powerful jaws and or by ejecting a white sticky repellent from an opening on their head. Soldiers can't feed themselves, they have to be fed by workers. Usually the number of soldiers is much smaller than the number of workers. Soldiers can be mandibulate or nosute, depending on the species. Therefore soldiers can be used for the identification of termite species. The lifespan of the soldiers is 1 to 2 years. Caste system. Caste system of termites. A. King. B. Queen. C. Secondary queen. D. Tertiary queen. E. Soldiers. F. Worker. Worker termites undertake the most labor within the colony, being responsible for foraging, food storage, and brood and nest maintenance. 58-59. Workers are tasked with the digestion of cellulose in food and are thus the most likely caste to be found in infested wood. The process of worker termites feeding other nestmates is known as trophallaxis. Trophallaxis is an effective nutritional tactic to convert and recycle nitrogenous components. 60. It frees the parents from feeding all but the first generation of offspring, allowing for the group to grow much larger and ensuring that the necessary gut symbionts are transferred from one generation to another. Some termite species may rely on nymphs to perform work without differentiating as a separate caste. 59. Workers may be male or female and are usually sterile, especially in termites that have a nest site that is separate from their foraging site. Sterile workers are sometimes termed as true workers while those that are fertile, as in the wood nesting Archodermopsidae, are termed as false workers. 61. The soldier caste has anatomical and behavioral specializations, and their sole purpose is to defend the colony. 62. Many soldiers have large heads with highly modified powerful jaws so enlarged they cannot feed themselves. Instead, like juveniles, they are fed by workers. 62. 63. Fontanelles, simple holes in the forehead that exude defensive secretions, are a feature of the family Rhinodermidae. 64. Many species are readily identified using the characteristics of the soldiers, larger and darker head and large mandibles. 59. 62. Among certain termites, soldiers may use their globular phragmotic heads to block their narrow tunnels. 65. Different sorts of soldiers include minor and major soldiers, and nosutes, which have a horn-like nozzle frontal projection, a nosus. 59. These unique soldiers are able to spray noxious, sticky secretions containing diterpenes at their enemies. 66. Nitrogen fixation plays an important role in nosute nutrition. 67. Soldiers are usually sterile, but some species of Archodermopsidae are known to have neotenic forms with soldier-like heads while also having sexual organs. 68. The reproductive caste of a mature colony includes a fertile female and male, known as the queen and king. 69. The queen of the colony is responsible for egg production for the colony. Unlike in ants, the king mates with her for life. 70. In some species, the abdomen of the queen swells up dramatically to increase fecundity, a characteristic known as physogastrism. 58. 69. Depending on the species, the queen starts producing reproductive elates at a certain time of the year, and huge swarms emerge from the colony when nuptial flight begins. These swarms attract a wide variety of predators. 69. Economic importance of Blattioidea. E.g., cockroach. Economic importance. Cockroaches are among the most cosmopolitan of all insect pests. They are associated with human dwellings throughout the world, hiding in cracks and crevices during the day and emerging at night to forage for food and water. Although they do not sting or bite, they are usually associated with unsanitary conditions and may carry a variety of human pathogens on their bodies. The decomposing remains of dead roaches and their feces are an important source of household dander that becomes airborne and provokes respiratory allergies in sensitive people. But cockroaches also have proven to be very useful as research tools, particularly for the study of insect physiology and toxicology. Major families. Blattidae includes the oriental cockroach Blatta orientalis and the American cockroach Paraplaneta americana. Bladelidae includes the German cockroach Blattella germanica. Bug bites. The American cockroach, Paraplaneta americana, is an introduced species that probably originated in Africa. Some cockroaches in the family Polyphagidae live as commensals in the nests of ants. Gromphodorina portentosa, a species from Madagascar, can force air out of its tracheal system to produce an audible hiss. This behavior is used to scare predators. These large, slow-moving cockroaches are sometimes kept as pets. Despite their bad reputation, only about a dozen species out of 4,000 worldwide are regarded as pests. The Serenum cockroach, Pycnoscelis serenamensis, is the intermediate host of a nematode, Oxysporum mansoni, that may cause blindness in poultry. Dipterans as vectors of diseases. Diptera is an important insect order as several species is known for medical, agricultural and veterinary significance. Most dipterans like mosquitoes and houseflies live closely to human settlement and cause severe problems including nuisance and transmit diseases to human beings. Mosquitoes as vectors. Mosquitoes can transmit diverse infectious pathogens and parasites that cause diseases such as dengue, Zika, chikungunya, West Nile fever, or malaria. In order to do so, though, a mosquito must bite a sick human first, thereby infecting itself with the pathogen. After some time, usually around 10 days, the mosquito itself becomes infectious. Mosquitoes can act as vectors for many disease-causing viruses and parasites. Viral diseases, such as yellow fever, dengue fever, and chikungunya are transmitted mostly by Aedes aegypti. This disease is spread through the bites of infected mosquitoes and cannot be spread person to person. The parasitic diseases collectively called malaria, caused by various species of Plasmodium is carried by female mosquitoes of the genus Anopheles. Lymphatic filariasis can be spread by a wide variety of mosquito species. Equine. Encephalitis viruses can be spread by mosquito vectors such as Aedes tularensis. Tularemia, a bacterial disease caused by Francisella tularensis, as well as arbovirus infections such as West Nile virus is variously transmitted, including by Culex. Zika, though rarely deadly, causes fever, joint pain, rashes and conjunctivitis. The most serious consequence appears when the infected person is a pregnant woman, since during pregnancy this virus can originate a birth defect called microcephaly. St. Louis encephalitis, a mosquito-borne disease that is characterized by fever and headaches upon initial onset of infection, arises from mosquitoes who feed on birds who are infected with the illness, and can result in death. The most common vector of this disease is Culex pipiens, also known as the common house mosquito. Heartworm disease, a parasitic roundworm infection that affects dogs is transmitted by mosquitoes. Adult heartworms infest the right heart and pulmonary artery, where they can cause serious complications including congestive heart failure. Musidae vectors. The common housefly is one of the most widely distributed insects and has the ability to transmit disease to people. E.g., cholera, typhoid, shigellosis. This fly is commonly found in homes and restaurants. Because of its close association with people and its ability to transmit disease, it is considered a greater threat to human welfare than any other species of non-biting fly. Houseflies can fly for several kilometers from their breeding places, carrying a wide variety of organisms on their hairs, mouthparts, vomitus, and feces. Parasites carried include cysts of protozoa, e.g., Entamoeba histolytica and eggs of helminths, e.g., Ascaris lumbricoides, Trichuris trichiora, and Enterobius vermicularis. Houseflies do not serve as a secondary host or act as a reservoir of any bacteria of medical or veterinary importance, but they do serve as mechanical vectors to over 100 pathogens, such as those causing typhoid, cholera, salmonellosis, bacillary dysentery, tuberculosis, anthrax, ophthalmia, and pyogenic cocci, making them especially problematic in hospitals and during outbreaks of certain diseases. Disease-causing organisms on the outer surface of the housefly may survive for a few hours, but those in the crop or gut can be viable for several days. Usually, too few bacteria are on the external surface of the houseflies except perhaps for Shigella to cause infection, so the main routes to human infection are through the housefly's regurgitation and defecation. A number of bacterial endosymbionts have however been detected, the greatest numbers being detected in the abdomen. Glossinidae vectors. The sisi are obligate parasites that live by feeding on the blood of vertebrate animals. Human African trypanosomiasis, also called sleeping sickness, is caused by Trypanosoma brucei. This disease is invariably fatal unless treated, but can almost always be cured with current medicines if the disease is diagnosed early enough. Sleeping sickness begins with a sisi bite leading to an inoculation in the subcutaneous tissue. The infection moves into the lymphatic system, leading to a characteristic swelling of the lymph glands. The infection progresses into the bloodstream and eventually crosses into the central nervous system and invades the brain leading to extreme lethargy and eventually to death. Animal trypanosomiasis, also called nagana, when it occurs in bovine cattle or horses or sura, when it occurs in domestic pigs, is caused by several trypanosome species. These diseases reduce the growth rate, milk productivity, and strength of farm animals, generally leading to the eventual death of the infected animals. The sisi fly vector ranges mostly in the central part of Africa. It is an order comprising of terrestrial insects with great powers of leaping and running, except in certain acrididae, flight is not well developed and alary organs are quite wanting in some species. Some families like Blattidae and Acrididae have aquatic species but they don't have any special structural modifications. E.g., Blattidae when submerged, keeps the abdominal apex in contact with the air. They are insects of comparatively large size. External anatomy. Head, hypognathous, rarely prognathous, frontoclypeal sutures are distinct, X-shaped tentorium without a central aperture. Framework of internal support within an arthropod head, formed by ingrowths of the exoskeleton, called apophyses, head widely attached to the thorax, free movement of head is limited. 1. Antennae, cetaceous, short or long. 2. Compound eyes, usually large and surrounded by band-like ocular sclerites, well-developed ocelli usually 3 in number, absent in apterous forms. 3. Mouthparts, well-developed mandibles, maxillary palps are 5-segmented and labial palps are 3-segmented. 4. Thorax, prothorax is large, pronotum extended backwards, meso and metathorax together form a strong pterothorax. 5. Legs, unequal in length, hindlegs with enlarged femora due to deposition of strong tibial levator muscles and adapted for jumping except in family like Grillotalpidae. Forelegs are modified into fossorial type with forotibia broad and toothed Grillotalpidae. In Grillidae, forotibia has proximally a pair of tympanal organs. 7. Wings, fore and hind wings are unequal, forewings are small, sclerotized, leathery. In Tedygonidae and Grillidae, forewings have stridulatory organs. Hindwings are membranous with large anal lobe folded fanwise 8. Abdomen, long and 11 segmented. Acrididae has a pair of membranous tympanal organs on either side of the abdomen. Cerci short and unsegmented except in Gryllidea. 9. Alimentary canal, straight or slightly convoluted, with large crop, gizzard-bearing teeth, midgut with anterior long cc, malpighian tubules are numerous forming an ampulla tetigonidae or opening into the gut Gryllidea. Hindgut has rectum with 6 rectal pads. Salivary glands are well developed with salivary reservoirs. 10. Nervous system. CNS has 4 to 6 abdominal ganglia. Sympathetic nervous system is well developed frontal and hypocerebral ganglia, a pair of esophageal nerves, corpora cardiaca, corpora allata and a pair of ingluvial ganglia. Thoracic and abdominal nerves and neurohemal egans are well developed. 11. Tracheal system. Open type with 2 pairs of thoracic and 8 pairs of abdominal ganglia. Trachea has segmental air sacs. 12. Circulatory system. Heart elongated with 5 pairs of abdominal ostia and 2 to 3 pairs of thoracic ostia. 13. Reproductive system. Female genitalia are well developed. Ovipositor has 3 pairs of long valves. Male genitalia is well developed composed of a pair of styles on 9th abdominal segment. Male has a pair of multifollicular testes, a pair of vas deferentia anteriorly modified into epididymis, a median ejaculatory duct, a pair of seminal vesicles and a median mushroom gland. Female has a pair of panoistic ovaries, a pair of oviducts open into a vagina, a spermatheca, a pair of accessory glands and a genital chamber. The clitorial glands are well developed in families like Blattidae which form highly developed ootheca. 14. Development. Eggs are elongate, laid singly or in clusters and are on the ground. The first instar nymph is vermiform per nymph. Total number of nymphal instars vary from 4 to 15. In apterous forms, metamorphosis is slight. Classification. Orthoptera has 2 suborders. 1. Encephera tetigoniidea, antennae longer than the body, tympanal organs on foratibiae, stridulatory organs on tegmina, elongated ovipositor, longhorned grasshoppers, bush crickets, mole crickets. The suborder has 3 superfamilies. A. Gryllocrydoidea, without stridulatory organs and tympanal organs but with long ovipositor. The families are Stenopelmatidae, Gryllocrydidae, Schizodactylidae, Rhapidophoridae. B. Tetigoniidea, long antennae, stridulatory and tympanal organs, long ovipositor, winged and wingless forms are seen. The families are Tetigonidae, Brophalongosidae. Circa. Grylloidea, males have stridulatory organs, 3 segmented tarsi, ovipositor needle-shaped. The families are Gryllidae and Gryllotalpidae. 2. Calyphora acridoidea, short-horned grasshoppers, locusts etc. Antennae shorter than the body, tympanal organs on the first abdominal segment, stridulatory organs on femur, ovipositor short or absent. The suborder has 2 superfamilies. A. Acridoidea, 3 segmented tarsi, well-developed ovipositor. Family Acrididae. B. Tridactyloidea, segmented vestigial tarsi, ovipositor absent. Family Tridactylidae and Cylindrochetidae. Family Tetigonidae, long-horned grasshoppers. Antennae are filiform, 30 or more segmented, extending far beyond the apex of the abdomen. Tegmina, leathery forewings slope obliquely backwards, membranous, left one overlapping the right and shorter than the wings. Stridulating organ found on the left tegmen. Right tegmen has a smooth transparent area of tense membrane which acts as a resonator for the tegmina. They make loud sound at night. A pair of auditory organs are present on each foratibia. Winged Tetigonidae are green and live among foliage, wingless forms are gray or brown in color and live in burrows, hollow trees, beneath stones etc. Ovipositor is longer than the body and used to lay eggs in the earth or in plant tissues in longitudinal rows. Eggs are not laid in ootheca. 5 to 6 molts are common in the family. Family Tetragidae, groundhoppers, pygmy grasshoppers. They are less than 20 millimeters in length with a long pronotum, which extends over the length of the abdomen sometimes to the tip of the wing. The first thoracic sternite is modified into a collar-like structure called sternomentum. They have scaly forewings and well-developed hind wings. Front and middle tarsi with 2 segments, hind tarsi with 3 segments. Auditory and stridulatory organs are absent. They show sexual dimorphism both in size and color. They have hemimetabolous development, in which eggs hatch into nymphs. Family Acrididae, locusts and short-horned grasshoppers. Antennae are shorter than the body and may be filiform or clubbed. There is a ridge formed of about 80 to 90 pegs on the inner surface of the hind femora which forms the stridulatory apparatus which is rubbed against the tough veins on the folded tegmina. The auditory organs are present on either side of the basal segment of the abdomen. Ovipositor is inconspicuous and has short valves with which they excavate a hole in the ground in which 80 to 100 eggs are laid in a mass. A gelatinous substance hardens to form a tough covering for the eggs. There will be 5 to 8 ecdysis in the life history and 2 generations can be seen in a year. They are voracious feeders of vegetation during their nymphal and adult stages. Locusts. Locusts belong to the subfamily Acridiinae, characterized by the presence of a long spine on the prosterna in between the anterior legs. The genus Locusta includes the world's oldest migratory locusts. It has 2 species, L. migratoria and L. pardalina. They form large swarms when the population of these locusts reach the peak. They migrate from their breeding ground, the exact reason of which is not known. The locust migration is not driven by hunger since during this period, they don't eat much but depend on their fat reserves. Cessation of flight is said to be due to internal physiological causes like maturation of gonads and connected to the discovery of suitable breeding grounds. The resulting progeny gives rise to Danica phase, which is not gregarious but aids in the distribution of the species. This phase in turn gives rise to migratory 1. Schistosera gregea is an example of locust that caused vast damage to vegetation in northern Africa, Iran, Afghanistan and northern India. There are two phases. A destructive migratory phase, Phasis gregaria, and a harmless solitary phase, Phasis solitaria. Two phases are connected by intermediates called Phasis transcens 1. Phasis gregaria. The nymphs are black, yellow or orange in color. On attaining maturity, the color changes especially in males. The adults have concave pronotum, prominently constricted. The wings have greater length than solitaria. Hind femur is shorter than forewing. Body temperature is high. Color change is shown after sexual maturity. Both adults and nymphs are gregarious. They are diaposing too. Phasis solitaria. Nymphs are extremely plastic in their coloration, changing the color according to the environment. The imago has arched pronotum without constriction. There is no change in the color of male and female. Hind femur is longer than forewing. Body temperature is low. They always behave as solitary grasshoppers. 3. Phasis transcens. Based on whether developing towards solitary or gregarious phase, they show tendencies of developing the characteristics of both gregaria and solitaria. The gregaria phase develops only in specific breeding areas, where a particular cobiontion of ecological conditions prevail. When the conditions are favorable, they form dense swarms, migrate to new breeding grounds, the resulting progeny develops into solitaria or transcens. Locust breeding and life history. Breeding of locusts koiniki with rainfall and eggs are laid in moist soil in the transitional areas between desert and semi-desert regions which provide biotic resistance to them. Life cycle has 3 stages: eggs, hopper and adult. Eggs are laid in batches of 150 or more in the burrow of 8 to 15 cm depth in sandy soil. Depending on the temperature, eggs hatch in 11 halves to 4 weeks to prenymph and after a few molts become hoppers. Structure and behavior in the hoppers changes to scattered or concentrated manner depending on whether they enter solitary or gregarious phase. After 3 to 10 weeks, final molt brings in the adult stage which survive for 10 to 15 days. There are 2 breeding seasons during a year, spring and summer, monsoon depending on whether the rainfall occurs in spring or summer in different breeding homes of the locusts. Migration of swarms. 1. Eastward swarms. From the winter rainfall areas of Middle East Baluchistan to summer rainfall areas like Sindh, Punjab, Rajasthan, Uttar Pradesh etc. during May to July. In the wake of monsoon rainfall, these swarms lay eggs and breed here till September-November if the rains continue and temperature is high. 2. Westward swarms. From September, many new swarms from monsoon breeding areas fly westwards into the spring breeding areas of Baluchistan, Iran, eastern Arabia where they breed again in the following spring. 3. Southward swarm. From Rajasthan to east and south damaging the crops of vast areas of India but without breeding, they die off. 4. Northward swarm. Overwinter in North India where they breed and damage crops of vast areas of India. Swarms stop only when the climatic conditions become unfavorable either in monsoon or spring in the breeding homes, else they multiply from season to season, year to year. Till favorable condition returns they remain solitary. Each swarm can contain a single mass of about 40 miles in length and several feet in height in which there will be 2,500 individuals. Swarms can spread to 2.5 sqkm with 100 million locusts which consume 300 tons of food while they settle. Biotic theory of periodicity of locust cycles Prodin, 1969. This explains probable causes of phase, transformation and swarm outbreaks in periodic and cyclic manner. Drastic climatic changes in the breeding areas are solely responsible for the periodic locust outbreaks, causing transformation of locusts from solitary to gregarious forms. It is said that when climate changes, natural enemies of locusts are reduced causing their population explosion. They start laying eggs extensively, absorb more solar heat to produce melanin in the integument and wing. Then for searching suitable environmental conditions, they collectively swarm and migrate to long distances. In normal climatic conditions, conversion of locusts from solitary to gregarious as well as swarming and distant migration never occurs. Locust control is done internationally by various organizations like Locust Warning Organization India, 1939 International Locust Research Center. Anti-locust operations under FAO. These organizations alert people about new outbreaks, swarms and suggest package schedule of locust control methods. Family Grylidae crickets. They resemble Tetigonidae in having long filiform antennae, exerted ovipositor, stridulatory organs on tegmina and auditory organs on tibia. They resemble Acrididae in having 3 jointed tarsi. Tegmina are folded flat over the abdomen, right tegmen overlying the left, bent abruptly downwards along the sides of the abdomen. The stridulatory organ comprises of a file and a hardened area called scraper on the tegmina against which the file of the other tegmen works. There is also a vibratory area or tympanum on both tegmina. During stridulation, the tegmina are elevated to 45° with respect to the abdomen, and moved back and forth so that the file of one tegmen is rubbed against the scraper of the other tegmen. This movement in turn vibrates the tympana which leads to the production of an audible sound. The cerci are exceptionally long and unsegmented, anal styli are short. Eggs are laid in the ground or in the pith of the twigs. The life cycle has 5 or more ecdyses. They are omnivorous, frequent hot dry areas and live in holes or burrows. Family Grylitalpidae, mole crickets. They are subterranean inhabitants seldom seen. The body is cylindrical in shape and covered by fine dense hairs. The head, prothorax and forelimbs are highly sclerotized, abdomen is rather soft. The forelegs are greatly inflated and modified for fossorial habit. The head bears thread-like antennae, compound eyes are much reduced. The 2 pairs of wings are folded flat on the abdomen, the hind wings are membranous, exceeding the tip of the abdomen in many species. In some the wings are reduced. There is no visible ovipositor. The females burrow into the soil to a depth of 30 centimeters and lays a clutch of 25 to 60 eggs. Females remain in an adjoining chamber to guard the clutch. Eggs hatch in a few weeks and nymphs are voracious feeders of plant material. The nymphs resemble the adults apart from the absence of genitalia and wings. It is a